Last night I had on an orange tie, so I had orange and blue, and I thought maybe the people from New England would say something about a football game that was held a little while ago between the Broncos and the Patriots. But since none of them brought it up, I thought I'd just bring it up. What the heck? But Dr. Hirsch gave me this green tie that I have on tonight. Okay, so the EGF receptor is not just something that gets mutated. This is a receptor and a growth factor that is used in the bronchial epithelium. If you look at normal bronchial epithelium, the basal cells express the receptor. And to turn over, when the adjacent cells make EGF, we can replace the bronchial epithelium. Now, this receptor is involved in the pathogenesis as well. If you look at dysplasias, there's very high expression in essentially all the dysplastic cells, whether there's angiogenesis associated with that or not. And as you know, the majority of lung cancers, especially squamous cancers, have very high expression. The mutations occur in adenocarcinoma, but squamous tumors have very high expression of the protein. And so there's a number of ways to deal with this. And of course, we know all about the TKIs. Whoops. We also know that this HER1 or EGFR heterodimerizes with a number of other partners that gives very strong signaling. So if I go back, there's a number of ways to interfere with that. And one, of course, is the development of antibodies that bind to the EGF receptor. So just to put things in context, this is the study of the ECOG, the famous ECOG study that led to the approval of Avastin. And at that time, I guess this improvement in the median survival of 10.2 months to 12.5 months was treated as something that has value. So Dr. Crawford last night told us all what value is. Now value in his definition is the benefit divided by the cost. Okay, so Avastin was expensive and there was a small benefit. But at that time, we sort of accepted that. Now, the first anti-EGFR antibody was Necitumumab, and it was humanized. So it was a murine antibody that was humanized. And the famous FLEX trial compared Necitumumab with a platinum-based doublet versus a platinum-based doublet alone. And in that study, the median survival with the control arm was 10.1 versus 11.3 with a hazard ratio of 0.87. So in terms of benefit, seem pretty similar to Avastin. Cost is, I don't know what the cost is. We'll talk about Necitumumab in a few minutes. But the FDA didn't like some of the trial design that was done in this study, and the drug wasn't approved. Now, if you looked by histology, there was benefit in both adenocarcinoma and in squamous carcinoma. Median survival went from 9 months to 10.2 months in squamous. Hazard ratio was 0.8 actually in the squamous patients, a little bit better than the overall study. Now, they also looked at protein expression, not gene copy number, but protein expression. And you can see that in the squamous cell patients, the median survival in the high IHC cohort was 8.9 months, versus 11.2 months in the cetuximab arm. So this suggested that there might be some patients that had more value, that is more benefit, at presumably the same cost. Whoops. So Dr. Hirsch lent me this slide. So should we go fishing and look at gene copy number in addition to protein expression? And the Colorado group, led by Dr. Garcia, developed a scoring classification, which included positivity, meaning that there was more than 40% of the cells having 4 copies or more, or frank gene amplification. So that was viewed as being positive. So that test was done in a series of Southwest Oncology Group trials, and this was, one could argue, not the best designed trial of all time, but this gave chemotherapy with cetuximab, followed by maintenance cetuximab, versus the sequential use of chemotherapy with maintenance cetuximab, and it didn't matter. The outcome was the same in both of these groups. But the SWOG looked at the FISH data from patients on both arms, and this shows the combination of results. And you can see that the positive patients had a much longer PFS and a much longer OS, whether the cetuximab was given at the start or as maintenance. And so that led to this somewhat infamous study called 0819, which was SWOG's randomized phase 3 trial looking at standard therapy or standard therapy with cetuximab. So in this trial, all histologies were included, and those that were BEV eligible got BEV. Those that were not BEV eligible, all the squamous patients just got chemotherapy with or without cetuximab. And it was a very large trial, and I'm not going to go through the results in the entire study, which was negative, but if one looks at those patients who had squamous cancer and were FISH positive, the median survival was 11.8 months in the cetuximab arm and 6 months in the control arm. And that was a hazard ratio of 0.56. Now, Dr. Crawford might think, geez, maybe there's more value here because there's more benefit here and the cost is the same. So what about combining cetuximab with radiation? Well, everybody knows this RTOG trial 0617 was sort of, in a way, double negative, showed high dose radiation was a bad idea, and it showed that in all comers, cetuximab didn't do anything. And unfortunately, RTOG was not as advanced as SWOG, so they didn't do FISH testing, although they did do IHC testing. And they found out that that in the patients that had high IHC, there was evidence that the cetuximab actually had a beneficial effect. Okay, so Eli Lilly, in their infinite wisdom, decided that many patients would get allergic to cetuximab because it was human, and there were a lot of first infusion reactions. And they also decided, I think, they could make more money with a human newer antibody. So they developed Necitumumab, which is a human, not humanized, monoclonal antibody to the EGFR. And they did 2 trials, one called INSPIRE, and that was in non-squamous. And they did another trial called SQUIRE, which was in squamous. And these gave standard chemotherapy with or without Necitumumab. Now, the INSPIRE trial was closed early by the Data Monitoring Committee because of some safety events, and we know that that study actually was negative. But the SQUIRE study completed its accrual and has been reported out. And these are the results of that trial, which was negative. And when they looked at the H-score, there was no evidence of particular evidence of benefit. This is the overall results of SQUIRE, and this is what Dr. Crawford wants us to focus on. In the overall trial, the gemcitabine cisplatin Necitumumab had a median survival of 11.5 months, and chemotherapy by itself was 9.9 months. So you can put your finger between these, but you can probably only put your pinky in there, not your thumb, because the benefit is relatively modest. Now, the trial met its primary endpoint. Primary endpoint was an improvement in survival. And based on that, the FDA approved Necitumumab for use. And so now we have this out there. And Suresh kindly gave me the figures, $11,000 a month. So over $120,000 a year. So we know the benefit and we know the cost. And so you can figure out what you think the value is. Now, what about those biomarkers that we talked about before? Does it matter if you have EGFR? You know, this was a company-designed study, and the primary endpoint was all comers. And they didn't have any hypothesis about any biomarker, but fortunately, Dr. Hirsch got them to do the biomarkers. So this was H-score, and you can see in terms of survival, like the FLEX trial, the survival was actually statistically significantly better, and the hazard ratio was 0.75 in the high group. But what about the FISH-positive group? So in the FISH-positive patients, the median survival was 12.6 months in the Necitumumab arm, and 9.2 months. That's a hazard ratio of 0.7. So now we have doubled, basically, the median survival benefit. So no matter how you score value, there's obviously higher value there. Now, there's not higher value for the company because not all the patients are FISH positive. As you can see, about half of the patients, a little less than half of the patients are FISH positive. But if you're thinking value, would you give this to everybody, or would you do a simple FISH test and select the patients most likely to benefit? Well, the problem with both 0819 and with SQUIRE was that these were not endpoints that were defined ahead of time. Both of the groups thought, well, let's do the biomarker, but none of them put it in their statistical analysis plan. So this is a hypothesis that's been generated. So who is going to prove the hypothesis? Basically, you need another randomized trial in FISH-positive squamous cancer to prove that this is the case. And it wouldn't have to be a huge trial because the benefit 0.7 is pretty good. Now, of course, another problem is what's going to happen if the PD-L1 inhibitors come along and beat chemotherapy? So that's going to make that trial more difficult to do. But I certainly would hope that the cooperative groups would think about doing such a trial. Maybe it would be stratified by PDL status, to show that you could have a benefit in both PDL high and PDL not so high. But if we want to have value, we got to do the trials, and I hope somebody does the trials. There are a couple other antibodies that are still being tested. Whoops, we heard from Merrimack earlier, and Merrimack has this EGFR antibody. We don't know exactly where it stands. Maybe the guy from Merrimack can say something about where the development of this stands. And then there's Necitumumab, which is another humanized EGFR antibody. So, see if I have a summary slide. Oh yeah, make sure that you don't schedule this meeting on these dates next year. And those of you that are hungry like me, in a very short time can go to dinner. That still could happen. I mean, there are many patients that I think the value is not enough. Okay, but there's probably some young, pretty fit patients with high EGFR copy number or a protein that I would seriously consider, treating. Now, the company, I can't speak for them, but they don't like that because they'd rather give the drug to everybody. But there's a lot of people in there that aren't getting much benefit, and there are some people that I think we can define that do have much more benefit, and it all makes scientific sense. The problem is they just didn't have that as a primary endpoint in the trial. No, it's about 40% of the squamous patients. So it's not most of them, but it's enough that you would treat. Yeah, still a lot of patients.
So that which just follows up. So if you had a patient right now with squamous cell came into your...
well, we do the FISH test, and it's right around the corner, so I would. I
would,
definitely would.
And then if it was positive, you didn't have a trial form, you would put it, so that would be how you would do it right now.
That's what I would do right now. I think it's totally reasonable. I mean, it's like HER2. Suresh, so hopefully ECOG will do the trial. Well, big swag on the trial.
Bad news is the NCI doesn't want any trial in squamous advance at this time for a variety of reasons. But follow-up question to what John asked. If there is a patient in the community that somebody wanted to do the FISH test, is there a commercial entity?
Abbott has the test is available.
And they would be able to use the Colorado definition.
Yep.